Hi, everyone. Thank you for having me. To start with disclosures, I'm a co-founder and employee of Ozette Technologies. The work I'll talk about today was done while I was a postdoc and data scientist in the Guettarda lab at Fred Hutch. So to set the scene, cytometry experiments can generate very complex data sets. Samples can be collected from multiple subjects, and samples are then labeled with marker panels to interrogate proteins of interest. After they've been labeled, samples are processed by an instrument to collect single-cell measurements on potentially millions of cells per sample. And depending on the experiment, large ones can generate hundreds of thousands of samples, and there can be noticeable biological or technical variation from sample to sample, which can make analysis challenging. So a typical analysis is a manual analysis called gating, where starting from the root population of cells, an investigator sequentially draws filters on pairs of markers to identify cells of interest. The target cells are typically known in advance, and this process can work well if you know what you're looking for. But as the number of possible protein combinations have grown exponentially with the number of markers, you can't look at everything manually anymore. So this invites a computational solution, but there are a lot of challenges there too. If you just think about clustering as an analog to manual gating, there are a lot of challenges you have to answer with any method, and some of those are listed on the screen. So in the course of the talk, I'll return to these points one by one to discuss how our method addresses these questions. So our method is called FAUST. It stands for full annotation using shape-constrained trees. It was inspired by a lot of the literature in forest-based algorithms, decision trees and random forests, as well as density-based clustering algorithms. The figure on the right is a schematic diagram from the publication, walking through the different steps of the method. But today, I'm going to show you what goes on under the hood when FAUST is applied to a real-world data set. The data set I'll use for this demonstration is from a study I was fortunate to be a part of, led by Florian Mayer and Jamie Erickson out of the Prelic lab at Fred Hutch. The study and data were published earlier this year, and the data are available on Flow repository. So among the data sets generated was a 30-parameter cytometry data set interrogating T cells and tumor samples from subjects with head and neck squamous cell carcinoma and site-matched oral mucosa tissue from healthy controls. And at a very high level, the investigators were interested in understanding differences between tissue and tumor samples measured in the cytometry data. So to look for differences, we applied FAUST to 22 samples stained by the T cell panel, 11 tissue and 11 tumor. The picture on the screen is from one of those samples, and in each subplot, you see the distribution of fluorescence intensity for a protein marker among live cells in that sample. Keep this picture in mind, since we'll be showing a lot of similar pictures over the course of the talk, where each subplot shows an expression distribution for a protein in a sample. And FAUST begins by exploring candidate gating strategies for each sample. And it generates candidates by first applying the dip test of Hartigan and Hartigan to each marker. The null hypothesis of the dip is that a marker will have unimodal expression distribution. So in this particular example, we fail to reject the null for the distributions in green, while we reject it for the 10 markers in purple. And when FAUST rejects the null, it then uses the tot string density estimator of Davies and Kovac to split markers into modal subgroups. The red thresholds in the pictures are determined by taking the center of antimodal components of the tot string. And FAUST uses those thresholds to start 10 trees for this sample, where each tree splitting the sample into subgroups of cells with expression measurements above or below the threshold for the given marker. So FAUST carries out the strategy for each sample independently, and it continues growing trees recursively in each sample, first applying the dip test, then using the tot string to split markers until it either fails to reject the null for all the markers in a subset, or it reaches depth 3 from the root. And using this forest, FAUST computes a score for each marker in each sample. Here I'm showing the score for 3 markers, CD4 in green, granzyme B in orange, LAG3 in purple. But for today, the most important thing to keep in mind is that the score is standardized to fall between 0 and 1. And to try to give you a qualitative sense of how to interpret these scores, here I'm showing a sample where granzyme B has a score close to 1. And when we look at the expression distribution, there's clear by modality. But if we switch over to another sample where the score is close to 0, you'll see that granzyme B has a unimodal expression distribution. So FAUST is not clear where to estimate a calling threshold. Consistently high scoring markers like CD4 in the experiment are typically well resolved across all the samples. When you look at the expression distributions, there's clear evidence of multimodality in all the samples. But consistently low scoring markers like LAG3 typically have unimodal expression distributions. So to answer FAUST's first question, FAUST's answer to the first question I listed earlier, which features should be used to cluster the data set. It's just to use consistently high-scoring features in a data set. Low-scoring features are set aside from subsequent clustering analysis. And in the example T-cell data set, these 7 markers were removed on the basis of their score. And for the selected markers that remain, adaptive thresholds are estimated for each sample. So for the CD4 shown here, you can see the adaptive thresholds in red, and they shift around from sample to sample depending on the location and scale. And Faust uses these thresholds to annotate clusters. So looking ahead, if a cluster Faust discovers has expression measurements for CD4 falling below the threshold in a sample, it's given the annotation CD4 negative. And if it falls above the threshold, it'll be given the annotation CD4 positive. So for the second question I posed earlier, how should you adjust for batch and technical effects in large experiments? Faust relies on its assumptions. The underlying assumption is that these batch effects will manifest as changes in the location and scale of expression distributions. And so there isn't actually explicit adjustment made to the methodology so long as that assumption is met. So here's a real world example of this occurring. My colleague Melissa Smith and I applied Faust as part of a reanalysis of a published ICS data set. And after reviewing the outputs, we found that Faust had automatically adapted to a batch effect in CD4 in the experiment. 4 samples are shown from that experiment here. The top 4 ridge lines are from one of the batches. The bottom 4 are from another. You can see both sample to sample variation, but also a shift to respect the batch. Returning to the T-cell case study, once markers have been selected and thresholds have been estimated for the selected markers, Faust then clusters each sample. It does this by randomly sampling partitions of each sample. The partitions are constructed by following the same recursive strategy of the dip test and the tot string that I described earlier, but the only difference here is that the depth constraint is removed. So by construction, each leaf of each tree in the schematic is going to have homogeneous expression distribution. Faust then assigns a quality score to each leaf of each tree, and finally selects a subset of leaves across trees in a forest for each sample, clustering the sample. And so each of these selected leaf nodes is then annotated relative to the adaptive threshold Faust had estimated earlier. What I'm showing here in the foreground, the green expression distribution is one of the selected clusters. In the background, the orange distribution shows the sample distribution among all live cells for these proteins. And this particular cluster gets the annotation CD103 negative because it falls below the CD103 threshold, CD28 positive because it's above the 28 positive threshold, and on for all the markers. Because Faust samples from the space of partition trees, and it does this procedure independently across all of the experimental samples, different annotations are observed with different frequencies across the experiment. Here's a CD19 positive annotation that was found in only one of the samples. The infrequency of this annotation group suggests it may be an artifact of the sampling process rather than biological signal. And here's another example of a CD4 positive T-cell annotation that was independently found in 11 of the 22 samples. And here's an example of a CD8 positive T-cell annotation that was seen in 20 of the 22 samples. So returning to the third question I posed earlier, how do you pick the number of clusters? Faust uses the following heuristic. It looks for an elbow in the curve describing how frequently annotations appear across experimental samples. And so here's a concrete realization of that curve. From it, you can see that almost 2,000 annotations appeared in one or more of the samples. But as you start demanding that annotations appear with more and more frequencies, there's rapid decay of the curve. And when you start to look at, say, 11 or more phenotypes or annotations that occur in 11 or more of the samples, only 50 annotations meet that criteria. And so the default estimate of the number of clusters is to estimate the elbow, and that value at the elbow is the number of clusters Faust will report. So for the final question, what kind of cells are in the resulting clusters? Well, by construction and conditional on the location of the thresholds, you can just read the annotation label to understand the composition of the cluster. So once you get the counts, you can do the usual downstream testing. In this particular study, we tested for the association with the tissue or tumor type and found several ICOS positive Treg subsets were significantly more abundant in tumor samples relative to oral mucosa. There are a lot more details about these types of cells in the publication, and I'll refer you there. We did a lot of benchmarking to characterize performance in our publication, both on simulated and reference data sets. The figure here is a subset of the results that we reported, where we tested Faust on simulated data sets where we varied the cluster structure ranging between 5 and 115 clusters in a given sample. And I see I'm coming up on time, so in brief, among the compare methods, Faust was able to well recover the cluster structure and also correctly assign abundances in terms of the adjusted RAND index. We've also applied Faust in a variety of other studies at this point, ranging from a Merkel cell carcinoma anti-PD-1 clinical trial, COVID-19 study, studying CD101 variants in HIV-1, and also have applied it to other reference benchmark data sets. So in conclusion, Faust is a method combining algorithms for market scoring, selection, clustering, and feature selection that can be used to discover and annotate phenotypes and high-dimensional cytometry data. So thank you all for your time, and thanks very much to my co-authors and collaborators.